Todos, Bom dia a todos, começou a gravar agora. Então, é com prazer que nós temos essa sessão hoje. O formato da sessão, a gente tem uma palestrante internacional que vai falar entre 40 e 45 minutos. Os chats serão usados, né? O chat será usado para postagem de perguntas e comentários e que depois serão lidos por mim e pela palestrante num debate para respostas. E, eventualmente, se alguém requisitar o áudio, basta colocar no chat e a gente dá um jeito de liberar o áudio para vocês falarem. E agora eu vou passar para inglês para apresentar a palestrante. So today it's an amazing pleasure to have Catherine Riou here. She's a very close friend. We work together in several projects, and she's extremely skilled in several techniques in immunology and advanced immunology and data analysis and how to visualize her data and to trace hypotheses and also to understand biology that underlies susceptibility to disease. So she is currently a senior research officer. She's an associate member of the Institute of Infectious Disease and Molecular Medicine in Cape Town, and also a Wellcome Center for Infectious Disease Research in Africa, the CIDRI Africa, and a contributing investigator at the University of Cape Town. So she's an immunologist, as I said, and her expertise is advanced flow cytometry. So she is a principal investigator at several grants with the EDCTP in Europe, NIH at the United States, Rosette Trees Trusts in UK, and research focused on cellular immune responses to HIV and also TB. But then in the past 2 years, due to the COVID pandemic, she pivoted her research interest towards understanding COVID-related questions, mainly investigated T-cell biology and T-cell responses during natural infection and also vaccination. So Catherine, thank you so much for accepting this invitation, and the floor is yours.
Hello, everybody. Good morning to you. It's the afternoon in Cape Town, and thank you very much, Bruno, for the kind introduction, and thank you very much for giving me the opportunity to present our SARS-CoV-2 research. For the rest of the talk, I'm going to switch off my video because we do have issue with bandwidth in South Africa. So the presentation that I'm going to give today will be divided in 5 sections. I'm going to talk a bit about the COVID. Sorry, I can't see my. Oh, yeah. I'm going to talk a bit about the COVID-19 epidemic in South Africa. I'm going to give a brief overview of the immune response to SARS-CoV-2, and then I'm going to present some data that we generated in Cape Town, looking at the T-cell response after infection and vaccination, looking at T-cell response to variants, and also looking at T-cell response in the context of HIV and TB co-infection. And most of the data that I will present today have been generated in collaboration with Dr. Wendy Berger. So this is the profile of the COVID-19 pandemic in South Africa. So South Africa experienced 4 very distinct wave of SARS-CoV-2 that were approximately 6 months apart. And during the last wave, there was a dissociation between the number of cases and the hospitalization and death. And this profile actually seems to be quite different to what you experience in Brazil, where your wave actually seems to actually overlap. So the official COVID data are reporting that approximately 3.7 South Africans have been infected with SARS-CoV-2, and more than 100,000 persons died. However, the COVID-19 death toll is really way more higher than what is reported. In a recent paper in The Lancet, people have actually estimated the excess mortality that is due to the COVID-19 pandemic. And while the global COVID-19 deaths reported is approximately 6 million, it is estimated that excess deaths could actually reach more than 18 million. And this actually maps show you the ratio of excess mortality to COVID-reported mortality. And you can see the highest ratio observed in Asia and in Africa, and also in Africa. And in South Africa, actually, COVID-related deaths are estimated to be 2 to 5 times more than the official reported number. So if we look a bit in more detail at the pandemic virological profile, you can see that each wave was characterized by a different SARS-CoV-2 variant, which actually became predominant very quickly. As you can see with that sharp actually swap from one variant to the other. So in South Africa, we had like the first wave was like ancestral SARS-CoV-2, then we had a beta wave, a delta wave, and then our last wave was a Omicron wave. And this profile also is quite different to Brazil, where it seems that you had multiple variants that coexisted for an extended period of time. So now looking at vaccine coverage, you can see on that map that there is major inequality between countries. And South Africa had sadly the lowest vaccination coverage. In South Africa, only 30% of the population is fully vaccinated. The first rollout of vaccines started in April 2001, and J&J was administered exclusively to healthcare workers. And the vaccination was open to the general population from July 2001, and Johnson & Johnson and Pfizer vaccine were offered to the population. And I think it's important to note that actually the prevalence of SARS-CoV-2 infection in South Africa is estimated to be 50 to 80%, depending on the province. So lots of people actually in South Africa have been infected by SARS-CoV-2. So now I'm just going to move on and give you a very, very brief overview of the immune response to SARS-CoV-2. So the immune response to SARS-CoV-2, as for many other viruses, engage all arms of the immune system. So when someone gets infected by SARS-CoV-2, they can generate antibodies, they will generate virus-specific CD4 helper T cells, and they also generate virus-specific CD8 killer cells. So those CD4 and CD8 T cells play a very different role. They can eliminate infected cells, they can stimulate humoral immunity, and they can also control the inflammatory milieu. So on top of all of these different cells, the CD4 response is also composed of like many different subsets that have distinct function. For example, there is like the T follicular cells that actually provide help to B cells. Some CD4 actually can assist the function of CD8 cells. Th1 cells also can provide antiviral activity, and some subsets such as IL-22 play a role in tissue healing. So overall, the control of the SARS-CoV-2 infection rely on a very complex and coordinated immune responses. So what are the mechanisms of protective immunity against COVID-19? So it seems to be quite clear now that high-level, long-lasting neutralizing antibody are very important for protection against SARS-CoV-2. We now know that actually SARS-CoV-2 is susceptible to neutralizing antibody. Antibody may be actually the only effector that can provide sterilizing immunity. Most of licensed vaccines have antibody as the mechanism or correlate of immunity. And a meta-study of several vaccine trials suggested that neutralizing antibody are predictive of protection. And as you can see on the graph on the right, vaccines that induce a high neutralizing antibody titer show the better, sorry, I can't move my, yeah, like you can see the titer here of in response to the vaccine. So the one that induce high titer actually show the highest efficacy. with the mRNA vaccine being the most efficient. But now there is also substantial evidence that actually T cells also contribute to the protection. So the presence of virus-specific CD4 and CD8 T cells have been associated with a better outcome in natural infection. It seems like quite clear now that a coordinated antibody, B cells, and T cell response is required for control. CD8 positive T cell provide partial protection in non-human primate model. And some clinical trials have shown that actually monoclonal antibody have modest impact on viral load. And also early induction of functional SARS-CoV-2 T cells associate with rapid viral clearance and mild disease. So all COVID vaccine have been shown to generate both antibody and T cell response, and the benefit of vaccination is now clear, both at an individual level, when there is a reduction of infection, there is a reduction of severe disease, hospitalization and death, and also a reduction of long COVID. At a community level, there is also the effect of vaccination with the reduction of infection and also a reduction of the period of infectiveness. But the efficacy and the benefit of vaccine will actually depend on the type of vaccine that is administered, the time since vaccination, the circulating SARS-CoV-2 variant, and also the immunological state of the recipient. So now I will move to the result section. And first, we're going to look at T cell response to SARS-CoV-2 after infection and vaccination. So for our study, we had access to several cohorts that were recruited during the pandemic, and the sample used in this for the result in this talk actually are coming from 3 different cohorts. The first cohort is a cohort of acute COVID-19 patients. So these patients were hospitalized, all of them. They had moderate to severe COVID. 56% were male, and the median age was like 52 years old. Those patients were recruited during the first wave, so when people were infected by the ancestral variant, SARS-CoV-2 variant. We also had access to convalescent patients. So these are actually healthcare workers that were sampled pre-vaccination. All these patients had either asymptomatic of mild COVID-19. None of them were actually hospitalized. They were predominantly female. The age, median age was 37 years old, and they were recruited during the first and second wave, so during ancestral SARS-CoV-2 and the Beta variant. And lastly, we also had access to a participant of the sessão trial, which was a J&J vaccine trial. That cohort was constituted exclusively of healthcare workers, and the patients included in that cohort were either uninfected, so they never had COVID, or they had either a prior episode of COVID or a breakthrough infection with COVID. But all the COVID episodes in those patients were either asymptomatic or mild. And that cohort had exactly the same distribution in terms of gender and age as compared to the convalescent, because these actually are the same patient. And in the J&J vaccinees cohort, the people that were infected, we had infection from all different waves. So with the 4 different prevalent SARS-CoV-2 variants that we experienced in South Africa. So let's move on to the results. And first I'm going to talk about the acute COVID cohort, so the hospitalized patient. So first we actually compare the clinical profile of SARS-CoV-2 patient and hospitalized control. So the hospitalized control were actually COVID negative patients that were recruited at the same time and in the same hospital. So to look at the clinical profile, we actually perform a clustering analysis of 8 clinical parameters that are known to be associated with COVID-19 severity. And this include the WH score, white cell count, CRP, D-dimer, ferritin, LDH, the magnitude of nucleocapsid-specific antibody, and the percent of affected lung that was assessed on X-ray. So on this graph, the COVID-19 patients are represented in red, hospitalized patients are in blue, deceased patients are depicted by a black square, and the people who survive are depicted by a gray square. So you can see that most COVID cases actually separate from a hospitalized control. And you can also notice that the patient who died tend to actually cluster together. So another way to look at the data is to actually do a principal component analysis, where you can see the separation between the COVID cases and the hospitalized control. And using the PC1 value from the PCA permits us to actually define COVID severity as a continuum. And as expected, the PC1 value, which reflects disease severity, was significantly higher in individuals who died compared to individuals who survived. So to measure T-cell responses, we used like flow cytometry, and we use a whole blood assay. So just very briefly, for that assay, we actually used 40 microliter of whole blood that was stimulated with SARS-CoV-2 peptide. The blood was incubated for 5 hours. And then after a series of manipulation, lysing the red blood cells and fixing the cells were stored at -80 for batch analysis. So for the flow panel, we used the 15-color panel, including T-cell lineage, the classic CD3, CD4, and CD8. We looked at memory differentiation, looking at CD45RA and CD27. We looked at activation with 4 different markers, CD38, HLA-DR, Ki67, and PD-1. We also measured the level of expression of granzyme B, which will give us an idea of the cytotoxic potential of these cells. And we measured 3 different functions, interferon gamma, TNF-alpha, and IL-2. So using that panel, we were able to track SARS-CoV-2 specific T cells. We're able to look at their polyfunctional profile, and we also were able to look at their phenotypic characteristic. So first, we looked at the frequency and the functional profile of SARS-CoV-2 T cell response in this patient. So the graph on the right presents the frequency of CD4 T cells on top, CD8 T cells at the bottom, in patients that were grouped according to their WHO status. So you can see that approximately 80% of the participants had a CD4 response, while the CD8 response was detected only in approximately 40% of the patient. But we didn't see any difference in the magnitude of SARS-CoV-2 T cell response, irrespective of disease severity. And that was the case both for CD4 response and also for CD8 responses. So the graph on the right here shows the polyfunctional profile of CD4 positive SARS-CoV-2 specific cells. And it shows basically the proportion of SARS responding cells expressing every possible cytokine combination, from polyfunctional cells expressing 3 cytokines to monofunctional cells. And you can see that with increasing disease severity, there was a progressive decline in the proportion of polyfunctional cells that was compensated by increase in the proportion of monofunctional TNF-alpha positive cells. So this association between severity and functional capacity was confirmed by the negative correlation that we obtained between the PC1 value, which is a reflection again of disease severity, and the proportion of polyfunctional CD4 specific cells. So next, we looked at the phenotypic profile of SARS-CoV-2 specific CD4 cells. So these graphs show you the R value between the phenotypic attribute of those cells and PC1 severity. And we found negative association with granzyme B and Ki67, and positive association with HLA-DR and TNF alpha monofunctional profile. So overall, this shows that actually SARS-CoV-2 specific T cells attributes, rather than their magnitude, associate with disease severity, with severe disease correlating with a poor polyfunctional potential, reduced proliferation and cytotoxic capacity, and enhanced activation. So next, we looked at the T cell response profile in convalescent patient. So to do so, we analyzed 74 healthcare worker who had mild disease, and the sample were obtained between 1 and 7 months post-infection and pre-vaccination. And you can see that as for the acute patient, the proportion of response was way higher in the C4 compartment compared to the CD8 compartment. We also show that the frequency of CD4 response was higher than the frequency of CD8 responses. And the CD4 response was highly durable, as we did not find any association between the frequency of CD4 responding cells and the time post-COVID diagnostic. So after 2 years of a pandemic, participant exposure history is becoming more and more complex. So we were wondering if repeated SARS-CoV-2 exposure, either through an infection or a vaccination, could affect SARS-CoV-2 immune response. So to answer that question, the patients were grouped according to the number of exposure they experienced. So for one exposure, it can be either one J&J vaccination or COVID episode.
Catherine, can you hear me?
Yeah, I can hear you. Was I on mute for the whole time?
I think, no, it's just when you started explaining this table, the upper table, it went muted. I don't know how.
Okay.
But, yeah.
I didn't touch anything. Do you want me to go back?
Yeah, can you repeat the description of
this? I'm really sorry, guys. I had no idea I was on mute.
It's okay.
Let me go back. Oh gosh. Oh,
no. We are good. All of that we heard.
You've done that? You've done that?
It's just the table of that slide.
Oh, the beginning of the table here.
Yeah.
Okay, that's good.
It was just 1 minute ago. It's fine.
Okay, that's good. Okay, yeah, gosh, thank you. Okay, so basically, to look at if basically repeated SARS-CoV-2 exposure affect the immune response, what we've done actually, we grouped our participants according to the number of exposure they experienced. So for one exposure, for example, it can be either a J&J vaccination or a SARS-CoV-2 episode. For 2 exposure, it can be 2 vaccination, a vaccination followed by a breakthrough infection, or a pre-infection after a vaccination, and so on for the 3 exposure group and the 4 exposure group. So first we look at the response, spike-specific IgG, so basically antibody response. And these graphs show you on top the proportion of responder to IgG, and at the bottom the magnitude of IgG, spike-specific IgG response. So you can see here that the second exposure lead to a very sharp increase both in the proportion of responder and also in the magnitude of the response. And the third exposure actually further enhance spike IgG magnitude. So the graph on the right show the response spike-specific CD4 response in blue and spike-specific CD8 response in red. And the profile were completely different. So after 3 exposure, there was an enhancement of the frequency of responder for both CD4 and CD8 response. But we didn't find any difference in the magnitude of T-cell response, irrespective of the number of exposure. So basically, this shows that antibody and T-cell response upon repeated exposure present very different dynamics. And that could suggest that actually the first exposure of SARS-CoV-2 that you get, either an infection or a vaccination, may dictate the imprinting of your T-cell response. So next, we wanted to know if repeated exposure could affect the polyfunctional profile of SARS-specific T-cells. So the graph on the right shows CD4 response and the polyfunctionality of these cells. So for CD4 response, repeated exposure led to a gradual increase in the proportion of interferon gamma positive IL-2 positive cells that was counterbalanced by the loss of TNF positive IL-2 positive cells. And when we look at the CD8 response after different exposure, we didn't see any change in the polyfunctional profile of these cells. So basically, this shows that while multiple exposure do not affect the magnitude of SARS-specific T-cell response, it may shape the functional profile of CD4 responses. So then our next question was to actually ask if the sequence of exposure, not the number of exposure, but the sequence of exposure will actually affect T-cell response. So to answer that question, we looked at the frequency of SARS-specific T-cells and the proportion of responder in our 3 different exposure groups. And each group was subdivided based on the sequence of exposure. So for example, for one exposure, the sequence of exposure can be either a vaccination or an infection. And the color coding here actually represent the infecting strain in each of the participants, being either ancestral, blue, beta infection in red, delta infection in tile, and Omicron infection in orange. So if you look at the 2 and 3 exposure group, you can see that the sequence of exposure does not impact the proportion of a responder or the magnitude of the response. However, for the 1 exposure group, infection seems to induce a more robust CD4 response compared to a 1 J&J shot with the higher proportion of responder and also with the higher magnitude of responses. So next, I'm going to move on talking about T-cell response and variant. So the emergence of successive SARS-CoV-2 variant raised an important question and basically asking, are variants recognized by immune response induced upon vaccination or due to a prior infection? So as antibody are first line of defense, several laboratories actually focused on measuring the ability of neutralizing antibody to cross-react with a different variant. And it is now clearly established that all SARS-CoV-2 variants evade neutralization from previous infection or vaccination. And this is nicely illustrated by these results from Becklitz that show that actually serum from convalescent patient infected by the Wuhan strain or serum from like a Pfizer vaccine participant show reduced neutralizing activity against all variants with Omicron being the most resistant to neutralization. And you see it's the same for actually serum from like Pfizer vaccines. But the good news is that like mRNA vaccine booster increase antibody cross-reactivity towards variant, including Omicron. And you can see on this graph that neutralizing activity against Omicron was restored after 3 doses of Moderna, 3 doses of Pfizer, and to a lesser extent in J&J vaccine that were boosted by Moderna. So being T-cell immunologists, we and others actually have focused on the T-cell response to SARS, and we ask a similar question: can actually spike-specific T-cell cross-recognize the variant? So in a paper recently published by TARC, they measure CD4 response to ancestral spike in 50 vaccine recipients, either Pfizer, Moderna, and J&J, and they compare that response to 8 different spike variants. And you can see that actually the T-cell response were very comparable regardless of the variant tested. And when they calculated the fold change between ancestral and variant, all the median were close to 1. And this shows actually that vaccine-induced CD4 response recognize all previous variant of concern or variant of interest. And they actually found exactly the same profile for the CD8 response. So we have done the same exercise with Omicron, and we compared ancestral and Omicron T-cell response in 40 vaccine recipients and 15 convalescent, either people that were infected with, vaccinated with 1 dose of J&J, 2 doses of J&J, or 2 doses of Pfizer. And we saw a modest but significant decrease in T-cell response to Omicron in every tested group. But overall, 70 to 80% of CD4 response induced upon vaccination or prior infection actually cross-recognized Omicron. So why are these T-cell response preserved despite mutation in spike? So to try to answer this question, we measure the contribution of variable epitopes to the overall spike CD4 response. So to do so, we use sample from a patient infected with the ancestral virus during the first wave. And the cells were stimulated with a peptide pool that is covering the entire ancestral spike, or a small pool of peptide containing only the beta variable epitope, but in their unmutated form, and the same peptide pool, this time containing the mutated version of this epitope. So we measure interferon gamma positive T-cell response to those 3 different pools. And as you can see, a bit more than 50% of the participants actually recognize the wild type version of the peptide covering the mutated region. And the median contribution of the wild type epitope compared to the full spike response was approximately 15%, ranging from 6 to 25%. In all the 12 responders that were able to recognize the wild type pool, that response was lost when we actually used the beta version of this peptide. So this actually shows that mutated regions are not always immunogenic. You can see like sometimes they are not recognized at all. And when they are recognized, they only modestly contribute to the overall spike response. So the cross-reactivity of T cells is likely due to the broad recognition of epitope against the entire spike protein and the preferential targeting of conserved region. And this is illustrated in the graph here, showing that spike mutation depicted in red for Omicron. So each line corresponds to a region where there is a spike mutation, and you have the spike mutation for Delta. And these actually mutation occur in regions that have low recognition prevalence by CD4. So that is represented by those gray area. So I will shift gear again and this time talk about SARS-CoV-2 T-cell responses in the context of other co-infection. So in countries with high HIV and TB burden, like South Africa and also like Brazil, the intersect of COVID, HIV, and TB raised like several concerns. So HIV, we all know that HIV induced profound alteration of the immune system with a dysfunction that often persists after ART. Thus, it could be possible that actually HIV infection can impair SARS-CoV-2 immune response, leading to unfavorable outcome. The COVID and TB interaction could even be more worrying. As you all know, like COVID-19, TB is a respiratory disease that elicits massive inflammation in the lung. And one can imagine that COVID and TB associated inflammation could actually have a synergistic effect. And each disease could fuel the other one. And on top of it, the profound lymphopenia and hyperinflammation that is induced during severe COVID-19 could actually maybe favor MTB reactivation in latently infected person. So in fact, in South Africa, active TB and HIV infection were associated with like COVID death, with hazard ratio of like 2.7 for active TB and 2.1 for HIV positive. However, age and uncontrolled diabetes were the main risk factor for unfavorable outcome as it was reported worldwide. So to study T-cell response in the context of other co-infection, we use sample from the hiatus cohort, which was the hospitalized COVID-19 patient that we obtained during the first wave of infection. And the participants were grouped according to their HIV and TB stages. So I just want to point out here that the number of co-infected patients that are included in this study were very limited, with only 7 participants who actually were COVID and active TB positive, and 8 patients who were triple infected with COVID, HIV, and were also active TB. So first, we looked at the impact of active TB and or HIV co-infection on SARS-CoV-2 specific CD4 response. So this graph shows you the frequency of SARS responding CD4 cells in the 4 different groups. And as you can see, HIV or TB alone did not really affect the proportion of SARS-CoV-2 responder. But in the HIV TB infected group, only 25% of the participant or the patient, including in that group, exhibited a SARS-CoV-2 CD4 response. But within the responder, the magnitude of the CD4 response was actually comparable. So when we look at the association between CD4 count and the SARS-CoV-2 immune response, we found that the CD4 count positively associates with the magnitude of CD4 responses, and also associate with the magnitude of nucleocapsid specific IgG. So these data actually suggest that severely lymphopenic patients may not be able to mount an adaptive immune response to SARS-CoV-2. So then we flipped, I guess I'm missing a slide, but that's okay. So then we actually flipped the question around, and this time asking what is the impact of COVID-19 on memory MTB specific CD4 response. So to answer that question, we used sample from 3 different cohorts. A pre-pandemic cohort that was recruited in 2018. We also look at our hospitalized control. So these are COVID negative, but were recruited at the same time as our COVID patient, and our COVID-19 cohort. So that graph shows you the MTB-specific CD4 T cell response in these different groups. And you can see that in LTBI patient, the magnitude of MTB-specific response is lower in COVID-infected individual compared to healthy control in the HIV infected group. And there was also the same trend observed in the HIV positive group. In the patient with active TB, we didn't find any change in the frequency of the MTB-specific CD4 response between healthy and COVID patient. So one can wonder actually if that decrease that we're seeing in MTB-specific cells is related to a COVID-induced lymphopenia that has been described in severe patient, and if such a default that we're seeing here could be normalized after COVID-19 recovery. So our last question was to define if COVID-19 can reactivate MTB-specific CD4 T cells. So we and other actually have previously shown that HLA-DR is a very robust marker to assess TB disease. And we've shown that actually elevated HLA-DR on MTB-specific CD4 cells is characteristic of active TB. So this graph here shows you the level of expression of HLA-DR on MTB-specific CD4 T cells in our different group. And you can see that in LTBI infection, HLA-DR expression was actually comparable. I mean, the HLA-DR expression, was not affected by acute COVID-19 infection, both in HIV negative and in HIV positive individual, suggesting that actually acute COVID-19 does not reactivate tuberculosis. So in summary, I hope that I managed to actually show you that robust CD4 and CD8 T cell response are generated in most infected person, with CD4 response being more prevalent than CD8. The quality more than the quantity of CD4 T cell response associate with disease severity. And in convalescent patients, T cell responses are maintained up to 9 months post-infection. Upon repeated spike exposure, that can be either an infection or a vaccination, antibody response are gradually boosted, while the T cell response present a very different dynamic with no major improvement in their frequency. And it seems like 3 exposures seems to be optimal for an optimal antibody and T cell response. Moreover, unlike neutralizing antibody, T cell response generated upon vaccination or prior infection are highly cross-reactive with variants, including Omicron. There is a high degree of T cell cross-reactivity between the high degree of T cell cross-reactivity between variants is likely due to the broad recognition of epitope and the preferential targeting of conserved region. We still have to define if actually breakthrough infection will lead to the generation of novel T cell response, targeting like the variable part of the virus. We also have to define how T cell response are maintained over time, longer than the 9 months post-vaccination. And also, it's still unclear, though, and it's still to be properly demonstrated if cell-mediated immunity contribute to a milder outcome that is observed in a breakthrough infection. And dealing with the co-infection, we're showing that actually control HIV does not seem to substantially alter SARS-CoV-2 CD4 response. However, patients with advanced lymphopenia are less able to mount a T cell or an antibody response. And during acute COVID, the frequency of MTB-specific CD4 response is reduced compared to pre-pandemic, but COVID-19 does not lead to TB reactivation with no alteration in the activation or the differentiation profile of the response. And I would like to finish by actually acknowledging like all the people that are involved in that work, either in South Africa, at the IDM, at ARI in Durban. A big thank you to Umtweko Mtusi, who actually led the recruitment of all the healthcare workers, all the people that are like leading the Sisanke trial, and and a big thank you also to Alexandre Sete, who actually was the one who was providing us with all the peptide that we actually needed to do those studies. Okay, I guess that's it for me, and I'm ready to take any questions.
Oh, thank you very much again, Kat. It was amazing. We have several questions here, and I have my own questions too. So I will start with the questions that were placed here at the chat, and there's someone, Mittermeier, which is an amazing investigator here as well, that I think he raised his hand, so he wants to speak. So, but let's first prioritize the people that placed their questions first. So Claudia Brodsky is a PI here, and she has her own lab, and she is doing a lot of amazing immunology-based studies in COVID as well. And she basically had this interesting question whether the preservation of T cells, the overall magnitude of the T cells, can we measure the ability of protection just by considering neutralizing antibodies, or just, you know, when we are assessing the effectiveness of a vaccine, should we really look into the CD4 cells or T cells, or just say, well, this vaccine is inducing a lot of antibodies, so that's what matters in the end? So what's your opinion about that?
So as I was showing at the beginning, there is a very nice correlation between vaccine inducing like high titer of neutralizing antibody and protection. So clearly antibody would protect you from infection and like to getting like SARS-CoV-2, but with the emergence of variants, you can see that those antibodies are able to escape the variant very quickly. So that's, I guess, when you're very happy to have a second line of defense. And as much as T cells are not going to protect you to get infected, they might protect you against severe disease and against like hospitalization, because these cells will be able to actually kill infected cells. So they won't protect you from infection, but they will probably protect you from like severe disease. So that's why I think it's important to measure both when you look at vaccine protection.
Oh, perfect. And then the second question is from another immunologist, and she works with leishmaniasis, but it's an amazing as well. And she has this question about whether you checked for regulatory responses, like, you know, the Tregs and the and if there is any influence in this subcompartment of immune response comparing recovered and diseased patients.
Yeah, that's actually a fascinating question. Unfortunately, we didn't look at Tregs and the production of like IL-10 or like potential. We didn't look really all in immune. Receptor inhibitor like PD-1, CTLA-4, like stuff like that. So basically, immune regulator. Yeah. We just look at the level of expression of PD-1. And I was quite surprised to see that actually like PD-1, I didn't show the data here, but like PD-1 is extremely high on both CD4 and CD8 T cells in acute infection. But that could be normal because you said the cells are just in acute phase, so they upregulate PD-1. But even in convalescent patients, we see actually very high level of PD-1 in those T cells. So I guess it's a very important point that need to be investigated to check if those CD4 T cells that are generated are actually like not, and I wouldn't call exhausted, but like regulated in the sense of their immune system, like their immune response is a bit dampened because of the expression of those immune regulator. Yeah, but I'm not talking about exhaustion. I'm more talking about these cells being regulated. Yeah.
But have you correlated the PD-1 with polyfunctionality to see if there is any there was any profile. There's something.
No, I haven't actually. Yeah, no, I haven't. I'll do that. If there's the correlation, because you see the difference that we see in polyfunctionality with severity. People can ask, is it the cause or consequences? I think it's not. It's just a consequences. It's not a cause. In a sense of like, because people are like with severe disease, they have higher viral load, SARS-CoV-2 viral load, and basically the cells are going to get higher inflammation, higher systemic inflammation. So the CD4 T cells are going to be like totally differentiated. So that's why they're less polyfunctional and they just like pump out TNF. But that's why I think it's very important to look at other subsets, as I was mentioning at the beginning of the talk, Treg, TFH, Th22. I mean, all those other like CD4 subsets that actually play a role in regulating the response.
So we have one more question from Patricia Veras, another immunologist and also works with experimental pathologists. And she is just saying that if you found this maximum response prediction and with the third dose of vaccination, what you predict for a fourth dose of vaccination, is that really necessary to boost for one more time?
So that's also like a very interesting question. As much as the boosting does not increase the frequency of your T cells, so we have to dissociate the boosting and the response to antibodies, where each boosting for antibodies seems to be quite beneficial because you increase the broadness of your antibody, you increase the magnitude of your antibody. Regarding T cell, boosting doesn't seem to increase the frequency of the response, but it could actually affect the memory differentiation of your response, the functionality of your response. So basically, generating maybe more tissue resident CD4 cells, altering their function more than their magnitude. And we need to do way more work actually to actually define if those successive boosts will actually affect what I call the quality of your T cell response more than the quantity. Because you can't, unlike antibody, you can always boost them. T cell response at one point, it will be pathological. If you keep increasing your T cell response, you need to reach a plateau, a certain equilibrium. But it's a very interesting question. I guess more research needs to be done to look at more the quality of those cells. So
we have a couple of more questions. Lucas Carvalho is also an immunologist, and he's asking whether there is any correlation between IL-2 and gamma double producers and the neutralizing antibodies. If you plotted a correlation between the T cell responses and also the antibodies in a single,
So actually you do find, we and others found the correlation between the frequency of CD4 response and the magnitude of antibody.
Good.
So there is a relationship between CD4 response and antibody response.
So then Tiago Graf from Fiocruz as well. So he has a question here, let me read. Do you think that the high prevalence of HIV in Southern Africa countries is a factor that can foster emergence of new variants? And since you show that patients with AIDS have more difficulty to assemble good immune response against it. So,
yeah, that's a very good guess, actually. And Alex Siegel in Durban, who's at ARI, they actually followed so they have a couple of cases, there's not a lot, but they followed like HIV infected individuals who are not virally controlled and who got SARS-CoV-2, and they actually were shedding SARS-CoV-2 for like months and months and months. And they sequenced actually the virus in those patients, and they could see actually there was evolution in viral evolution, SARS-CoV-2 viral evolution in that patient. So at one point it was said, people with like lymphopenia could actually promote the emergence of a new variant. But yes, that's a very good point.
Yeah, that's for future studies as well. So, and then we have Manuel Barral from Fiocruz. Although T cells do not protect against infection, spike protein-based vaccines are protective. So he's asking what epitopes may be involved in protection.
For antibody or for T cells?
For T cells, yeah.
Oh, so the T-cell response to, so first of all, your T-cell response is not exclusively to spike. You have a response to nucleocapsid, to the membrane protein, to other part of the virus. And the T-cell response to spike and most of these protein actually covered a lot of, almost the entire, it spanned along the entire spike. So there's lots of regions that are immunogenic. To determine specifically which one will be the most prevalent or the most important for protection, it will be difficult to tell, because depending on patient HLA, your specific HLA, you might not mount a response to exactly the same epitope. That's going to be conditioned by actually the HLA class 1 and class 2 that you are carrying. So, but the good news is like your T-cell response seems to be able to recognize spike, like a lot of different epitope on spike, and also a lot of different epitope in the membrane and in the nucleocapsid. We not often, I mean, I haven't shown any data about that, but we've done a lot of work also on like N and M responses, and these T-cell response also play a role, not just the spike specific T-cell response.
That's amazing. So we have a lot of students here. So I have a technical question for you. And most of them work with flow cytometry. So you performed a very nice assay with whole blood. And I know this because I've been working with you, but you do a whole blood stimulation assay for 5 hours, and then you fix the cells, and then you freeze them. So then in order to have good staining, and your plots are fantastic, do you have any tips on how you need to choose exactly which antibody clones can how, just tell us a little bit about your experience on that.
So I'm a huge fan of whole blood assay. There's like several advantages and several inconvenience. The main inconvenience is because we fix our cells, we cannot go back and change our stimulation. Okay, so once you decide you stimulate your cells, like you cannot actually go back. It's not like PBMC where you can actually say, oh, there's a new variant, let's stimulate the cells with a new variant. But the advantage of like whole blood assay, first, it's like it's quick. And the whole blood also preserve the media, you know, like you have all your cell type here. So we have way better response. I've done a comparison between PBMC against whole blood assay and using the same stimulation, and you have way bigger response with whole blood assay. But because we fix the cells right after stimulation, there's a couple of problems. So you cannot look at any markers such as like chemokine receptor, because those markers are very sensitive to fixation. So basically it's impossible to look at chemokine receptor. Most of the markers that I'm using, so funny enough, some clones don't work, others do. If you want, I can send you a list of like clones that don't work. So, you know, like instead of reinventing the wheel. But most of the time, except chemokine receptor, most of the time, like the other markers are quite robust, and it's quite easy to actually detect them. And I'll happily share my protocol and also my panel if you need to.
Just a revision here. We have just a very few minutes left. Bahal was referring to actually the RNA-based vaccine-induced protection without any other protein there, right? So when he asked whether
okay, yeah, because you just have spike obviously in the vaccine. So I don't think people really looked in detail in like actually looking at the breadth of the response in response to a vaccine. I haven't seen any publication looking at that. But that will be very important to see in response to either natural infection or vaccination, if you generate the same type of breadth between for spike, you know, infection with spike or vaccination with spike. I guess people have been limited by the number of cells that we are, you know, that we have to actually do such study, because you will need to actually go almost peptide by peptide or like do matrix and then deconvolute those matrix. Yeah. So, but I think it will be very important to have a better understanding of which region of spike are actually targeted in the context of like vaccination.
So then we have another question. It's not a question, it's just a matter of like if the person understood or not. Paulo wrote that the TCL response seems to be more robust from natural infection compared to vaccination. Is that correct? Is that he's just asking if he understood correctly?
Yeah, that's correct. That's the data that we got, but we were comparing one dose of Johnson & Johnson compared to like one infection. So as much as Johnson & Johnson was like started as like a single dose vaccine, now like they give 2 doses of Johnson & Johnson, so that we need to look at that. But it seemed that actually, yes, infection give a higher proportion of responder and higher magnitude of response. But is magnitude the key? You know, like is magnitude the key of like protection, or is it like the quality more than quantity question? But yes, that's the data we're showing. When I say better protection, it's more robust in terms of prevalence and frequency.
Okay. Then we have Mittermeier. Mitter, you can open your phone. Yeah.
First of all, congratulations. It was really a fantastic presentation.
Thank you.
My question, I have basically 2 questions. The first one is, have you had the opportunity to observe any correlation between T-cell immune response outcome with the initial innate immunity type, immune response profile? If you had observed, if this had correlation with gender and obesity.
Okay.
And the second one, when we start with this pandemic, we had the opportunity to say, oh, I had less cases of COVID in Africa. Is this connected because people had TB or they're vaccinated by TB? So my second question is to be vaccinated for TB or to have TB but to be cured, this works to protect against severe outcome in COVID?
Okay, so your first question, if I understand properly, correctly, is like the relationship between innate response and active response. Am I correct?
The innate response and the outcome of the T-cell response.
So
I'm sorry, and also if you had the opportunity to look if those individuals were or not exposed to others beta coronavirus.
Okay. So let me slice that. So I'm a T-cell immunologist. I don't do innate at all.
Yeah, when we work together, I do the myeloid studies,
yeah.
and she does the you know, the
that's Bruno's work. It's not mine. So I won't be able to answer that. I won't be able to answer that question. Then the question was about like, is there a difference between gender and
obesity?
And obesity. So age and chronic condition, all of that have been shown, like obesity, diabetes, inflammatory disease have been shown to actually lead to like a weakened vaccine response in some people. You know, like people don't respond as well to vaccine, like so that could clearly have an influence of like the outcome for that. Gender, it's true that there was a dissociation. You know, more male actually died of COVID compared to female. I haven't looked in detail in our, and you can see our COVID cohort, like acute cohort, it's mainly 60% are actually male. I haven't looked in detail at the difference between male and female, but like that would be something good to check if, but yeah, no, I haven't looked at that. And your last question was regarding vaccination. Should you get like
if to be exposed to TB or to get TB vaccination, if it is alter the T-cell response outcome?
So if previous infection will alter your vaccine
TB.
outcome? Oh, TB.
TB.
Oh, sorry. Sorry, like I'm like, you know, I'm French, so. I don't know if like, you mean TB vaccination, if people with BCG who got BCG vaccine.
Yes, that received BCG.
Okay, so there was a couple of like study that looked at vaccinating people with BCG, hoping that it's gonna actually help. I'm not sure the data is very convincing, to be honest, or strong enough to actually strongly say yes, it's worth it to get additional BCG vaccination. We know that BCG actually vaccination protect kids from other disease, but when you're an adult, I'm not sure if it's gonna be the same. Sorry, I'm very unclear in my response, but actually, basically, I don't know.
Thank you very much. Congratulations for a very nice presentation.
Thank you very much.
Oh, thank you.
Thank you, Bruno.
Thank you, Mittermeier. So I think that's it, Kat. And I hope to receive you here so you can visit us and teach more about this like amazing skills in cellular immunology. I'm getting all private messages, like, ask her to send a panel of antibodies that is working.
Yeah, no, I believe in like sharing in science 100%, and I hate the idea of reinventing the wheel and having to re-optimize stuff that is already optimized. I'm in the lab, so I know exactly the time it takes. So Bruno, don't hesitate to actually share my email with like people, and I will gladly share my protocol and my panels.
Oh, thank you so much, and we see you soon. Thank you so much, guys.
Okay, bye everybody. Bom dia.
Pessoal, com isso a gente encerra a sessão. Obrigado a todos. Só um recado para vocês. Vai ter agora a apresentação do Prêmio Gonçalo Muniz. Eu não sei se a Natália está online e poderá falar um pouco mais detalhes, mas a gente está aí, Natália. Então, Natália, é com você.
Obrigada. Parabéns pela sessão, hein, Bruno? E Catherine.
Okay.
Oi, gente. Então, bom dia a todos que estão aqui no Zoom.